Hello everyone, welcome to this detailed session on red blood cells, the microscopic powerhouses responsible for delivering life-sustaining oxygen to cover every corner of your body. Today we will break down their structure, functions, lifespan, and clinical relevance in a simple, visual, and exam-focused way. By the end of this lecture, you will not only understand RBC's physiology, also, you will be able to connect it to a real life conditions like anemia and jaundice. So grab a pen and paper. Let's dive deep into the bloodstream. Let's begin by understanding the structure of red blood cells or RBCs. These cells are highly specialized for their role in gas transport. The first thing to notice is their biconcave disc shape. This design is not random. It significantly increases the surface area to volume ratio, which is ideal for rapid oxygen and carbon dioxide exchange. Another important feature is that RBCs have a very flexible membrane, allowing them to deform easily and pass through even the narrowest capillaries. This flexibility is due to the special proteins within their membrane and cytoskeleton. Unlike most cells in their body, mature RBCs have no nucleus or mitochondria. The absence of a nucleus creates more space for hemoglobin, the molecule that carries oxygen. And without mitochondria, the cell avoids consuming oxygen for its own metabolism. Instead, it relies on anaerobic glycolysis to meet its energy levels. Now let's talk about the cell membrane. It's composed of a lipid bilayer containing phospholipids and cholesterol that provide both flexibility and durability. Embedded in this membrane are essential proteins, most notably band 3 protein, which helps in exchanging bicarbonate and chloride ions during carbon dioxide transport, and glycophorins, which stabilize the structure and prevent clumping of RBCs. So beneath the membrane or this membrane lies a cytoskeletal network of spectrin and actin. This framework maintains the shape and mechanical integrity of the red blood cells. And finally, the key functional molecule inside RBCs is hemoglobin. It has four globin chains, two alpha and two beta chains in adult hemoglobin. Each chain is attached to a heme group containing iron, which binds to oxygen. So one hemoglobin molecule can carry up to 4 oxygen molecules. There are also minor forms like HbA2 and fetal hemoglobin HbF also, which has higher affinity for oxygen to support fetal development. Now that we have seen how RBCs are built, let's explore what they actually do. The primary function of red blood cells is oxygen transport. In the lungs, where the partial pressure of oxygen is high, hemoglobin binds to oxygen and forms oxyhemoglobin. This oxygen loaded hemoglobin travels through the bloodstream to the body tissues. There, under low oxygen conditions, it releases oxygen and becomes deoxyhemoglobin. But red blood cells also play a significant role in carbon dioxide transport, which is just as important. About 23% of carbon dioxide binds directly to hemoglobin, forming carbaminohemoglobin. However, the majority, around 70%, which is carried as bicarbonate ions. This transformation is catalyzed by the enzyme carbonic anhydrase, which converts carbon dioxide and water into carbonic acid, which then dissociates into bicarbonate and hydrogen ions. This leads to another critical role of RBCs, maintaining acid-base balance in the blood. Hemoglobin acts as a buffer by binding to excess hydrogen ions, which keeps the blood pH stable around 7.4. So, in the essence, red blood cells not only deliver oxygen but also help in removing carbon dioxide and maintaining pH homeostasis. Red blood cells have a finite lifespan, about 120 days. After this period, they begin to wear out due to constant oxidative stress and damage to their membrane proteins. Since they do not have a nucleus or mitochondria, they cannot repair themselves. As RBCs age, they lose flexibility and start showing markers of senescence, like exposed phosphatidylserine, which act as a signal for their removal. These aged cells are mostly destroyed in the spleen, but the liver and bone marrow also contribute to this process. The spleen plays a major role. It filters out old, rigid RBCs that can no longer pass through its narrow capillaries. Inside the macrophages of these organs, RBCs are broken down. The globin chains are broken into amino acids and reused for protein synthesis. The heme protein is converted to biliverdin, then to bilirubin. Unconjugated bilirubin binds to albumin in the blood and is transported to the liver, where it is conjugated and excreted into bile. In the intestine, it is transformed into urobilinogen, which contributes to the color of the feces and urine. Iron from heme is not lost, it's recycled. It binds to transferrin in the blood and is either stored in the liver as ferritin or sent back to the bone marrow to help make new RBCs. When this process is disrupted, clinical conditions arise. Excess bilirubin in the blood leads to jaundice, and excessive destruction of RBCs may lead to an enlarged spleen known as splenomegaly. Let's conclude by reviewing the most important points about RBC or red blood cells. Their biconcave shape is not just structural, it is functionally vital for maximizing gas exchange. Inside, hemoglobin serves as the core molecule for transporting both oxygen and carbon dioxide. RBCs have no nucleus or mitochondria, meaning they dedicate almost all their space and energy to gas transport. Their flexible membrane allows them to reverse through even the tiniest blood vessels, and they live for more about 120 days, after which they are systematically broken down and their components, especially iron, are recycled. Their destruction takes place mainly in the spleen, but also in the liver and bone marrow. If you found this video helpful, like it, share it with your friends, and make sure to subscribe the channel to get more content like this. Thank you for your patience.